So today we will be discussing on aerobic respiration, anaerobic respiration and bacterial fermentation. So all living cells, they depend on biochemical reactions to maintain homeostasis. So metabolism is the total of all the chemical reactions that occur in the cell. Metabolism is also classified into two parts, that is catabolism and anabolism. So in catabolism, large and complex molecules are broken down into smaller, simpler molecules with the release of energy, while in anabolism, it involves the synthesis of complex molecules from simpler ones with the input of energy. Microbes can be metabolize a wide range of organic as well as inorganic matter, from complex organic molecules like cellulose to inorganic molecules and ions such as atmospheric nitrogen, molecular hydrogen, sulfide, manganese ions, ferrous ions and ferric ions. By metabolizing such substances, microbes chemically convert them to other forms. In some of the cases, they produce chemicals that can be harmful to other organisms, whereas in others, it produces substances that are essential to the metabolism and survival of other life forms. So now we'll first see aerobic respiration. So aerobic respiration is the aerobic catabolism of nutrients to carbon dioxide, water and energy. And it involves an electron transport system in which molecular oxygen is the final electron acceptor. For most of the prokaryotes and eukaryotes, they use aerobic respiration to obtain energy from glucose. Microorganisms, they employed several metabolic pathways to catabolize glucose and other sugars. They degrade this sugar to pyruvate and other similar intermediates by three different routes, that is glycolysis, pentose phosphate pathway and Ettner-Dordorf pathway. In glycolysis, this glucose is phosphorylated twice and eventually it is converted to 1,6-bisphosphate. In this stage, it does not yield energy, but two ATP molecules are expanded for each glucose molecule. The enzyme fructose 1,6-bisphosphate aldolase, it catalyzed the cleavage of fructose 1,6-bisphosphate into two halves, each with a phosphate group. So one of the product, that is glyceraldehyde 3-phosphate, it is converted directly to pyruvate in a 5-step process. The dihydroxyacetone phosphate can be easily changed to glyceraldehyde 3-phosphate. So this glyceraldehyde 3-phosphate is first oxidized with NAD as the electron acceptor, and a phosphate is simultaneously incorporated to give a high energy molecule called 1,3-bisphosphoglycerate. The high energy phosphate on the carbon 1 is subsequently donated to ADP to produce ATP. This synthesis of ATP is called substrate level phosphorylation. The phosphate group in the 3-phosphoglycerate, it shifts to carbon 2 and 2-phosphoglycerate is dehydrated to form a second high energy molecule, that is phosphoenol pyruvate. This molecule, it donates its phosphate to ADP, forming a second ATP and a pyruvate. So the glycolytic pathway, it degrades one glucose to two pyruvates. The second pathway, that is the pentose phosphate pathway, it can take place either aerobically or anaerobically. This pathway, it starts with the oxidation of glucose 6-phosphate to 6-phosphogluconate, followed by the oxidation of 6-phosphogluconate to the ribulose 5-phosphate and a carbon dioxide. So NADPH is produced during the oxidation. Further, ribulose 5-phosphate is then converted to a mixture of 3 through 7 carbon sugar phosphate. So the overall result is that 3-glucose 6-phosphate, it is converted to 2-fructose 6-phosphate, glyceraldehyde 6-phosphate and the 3 carbon dioxide molecules. Further, the intermediates that are formed in this pathway can be used in two ways. That is, fructose 6-phosphate can be changed back to glucose 6-phosphate, while glyceraldehyde triphosphate is converted to pyruvate by the use of glycolytic enzymes. And this results in the complete oxidation of glucose 6-phosphate to carbon dioxide and the production of a great deal of NADPH. The next pathway is the third pathway, that is the Ettner-Dordorf pathway. And this pathway, it starts with the formation of glucose 6-phosphate and 6-phosphogluconate. Further, this 6-phosphogluconate is dehydrated to form 2-keto-3-deoxy-6-phosphogluconate. So this 2-keto-3-deoxy-6-phosphogluconate is then cleaved by the 2-keto-3-deoxy-6-phosphogluconate aldolase to pyruvate and glyceraldehyde triphosphate. The glyceraldehyde triphosphate, it is then converted to pyruvate via the glycolytic pathway. This pathway, it yields 1 ATP, 1 NADPH and 1 NADH per glucose metabolized. The Ettner-Dordorf pathway is found in Pseudomonas, Rhizobium, Azotobacter, etc. Now we'll come to the formation of acetyl-CoA through the transition reaction. The transition reaction, it connects the glycolysis to the citric acid cycle, which is also called the Krebs cycle, through a process called oxidative decarboxylation. So this transition reaction, it converts the 2 molecule of 3-carbon pyruvate from glycolysis into 2 molecules of the 2-carbon molecule acetyl coenzyme A, or which we commonly call it as the acetyl-CoA, and 2 molecules of carbon dioxide. The 2 molecules of acetyl-CoA, it then enter the citric acid cycle. The 2 NADH molecules that are produced carry electrons to the electron transport system for further production of ATPs by the oxidative phosphorylation. Now we'll come to the Krebs cycle. The substrate for the Krebs cycle, also known as the TCA cycle or the tricarboxylic acid cycle, is the acetyl-CoA. In the first step, acetyl-CoA is condensed with a 4-carbon intermediate, that is the oxaloacetate, to form citrate and to begin the 6-carbon stage. Further, this citrate is rearranged to give the isocitrate. Then isocitrate is subsequently oxidized and decarboxylated twice to yield alpha-ketoglutarate, then forms the succinyl-CoA. At this point, 2 NADH are formed and 2 carbon are lost from the cycle as the carbon dioxide. Then the cycle now enters the 4-carbon stage, during which 2 oxidation step, it yields 1 FADH2 and 1 NADH per acetyl-CoA. In addition, 1 GTP is also produced from succinyl-CoA by substrate level phosphorylation. Eventually, oxaloacetate is reformed and is ready to join with another acetyl-CoA. Thus, TCA cycle, it generates 2 carbon dioxide, 3 NADH, 1 FADH2 and 1 GTP for each acetyl-CoA molecules that are being oxidized. So TCA cycle enzymes are widely distributed among the microorganisms. The cycle is functional in many aerobic bacteria, free-living protozoa, and most algae and the fungi. So now we'll come to the electron transport chain. The electron transport chain of bacteria, it operates in the plasma membrane. So most of the ATP that are being generated aerobically, it comes from the oxidation of NADH and FADH2 in the electron transport chain. So electron transport chain is composed of a series of electron carriers that operates together to transfer electrons from donors like the NADH and FADH2 to acceptors such as oxygen. The electrons, it flows from the carriers with more negative reduction potential as to those with the more positive potential, and eventually it combines with oxygen and hydrogen ions to form water. Bacterial electron transport chain, they vary in their electron carriers, example in their cytochromes, and are usually extensively branched. Electrons often enter at several points and they live through several terminal oxidases. Bacterial electron transport chains are usually shorter and possess lower phosphorus to oxygen ratio than mitochondrial electron transport chain. The coenzyme Q, which is also known as ubiquinone, it carries an electron and donates them to both the branches. But the branches it operate under different growth condition. The cytochrome D branch shows very high affinity for oxygen and it operates at low oxygen level, usually when the bacterium is in the stationary phase of growth. So this branch is not as efficient as the cytochrome O branch because it does not actively pump the protons to the periplasmic space. The cytochrome O branch, it shows moderately high efficiency for oxygen and it operates at high oxygen concentration. This branch operates normally when the bacterium is in the log phase of its growth, that is when the bacteria is actively growing and actively pumps the protons in the periplasmic space. So now we'll come to the anaerobic respiration. The anaerobic energy yielding process in which the electron transport chain acceptor is an oxidized inorganic molecule other than the oxygen is termed as the anaerobic respiration. Here, the major electron acceptors are the nitrate, sulfate and carbon dioxide. Some of the bacteria can use nitrate as the electron acceptor at the end of the electron transport chain and produce ATP. Such process is called the dissimilatory nitrate reduction. So now we'll see nitrate reduction. Some microbes, they are also capable of using nitrate as their terminal electron acceptor. The electron transport system used is somewhat similar to the aerobic respiration, but the terminal electron transport protein, it donates its electron to nitrate instead of oxygen. Nitrate reduction in some species such as E. coli is a 2-electron transfer where nitrate is reduced to nitrite by nitrate reductase, which replaces the cytochrome oxidase. Now we'll see denitrification. So denitrification is an important process in agriculture because it removes the nitrate from the soil. So nitrate is a major source of nitrogen fertilizer in agriculture. Almost 1/3 the cost of some types of agriculture is in nitrate fertilizer. The use of nitrate as a respiratory electron acceptor is usually an alternative to the use of oxygen. Therefore, soil bacteria such as Pseudomonas and Bacillus use oxygen as the electron acceptor if it is available and does not use nitrate. E. coli utilizes nitrate as well as fumarate as the respiratory electron acceptor, and so it may be able to continue to respire in the anaerobic intestinal habitat. Nitrite, the product of nitrate reduction, is a highly oxidized molecule and they can accept up to 6 more electrons before being fully reduced to nitrogen gas. Microbes such as Paracoccus species, Pseudomonas strugeri, Pseudomonas aeruginosa and Rhodobacter sphaeroides are able to reduce nitrate to nitrogen gas. So this process is carefully regulated by the microbe since some of the products of the reduction of nitrate to nitrogen gas are toxic to metabolism. So now we'll come to fermentation. So fermentation is the enzymatic decomposition and utilization of foodstuff, particularly carbohydrates, by microbes. It is also an energy-yielding process in which organic molecules serve as both the electron donors and the acceptors. In microbial fermentation, NADH is oxidized to NAD and the electron acceptor is either pyruvate or a pyruvate derivative. There are different types of fermentation. First, a type of fermentation known as alcoholic fermentation. It involves the conversion of sugar to ethanol and the carbon dioxide. In this type of fermentation, pyruvate is decarboxylated to acetaldehyde, which is then reduced to ethanol by alcohol dehydrogenase with NADH as the electron donor. Further, lactic acid fermentation, it involves the reduction of pyruvate to lactate. Here, lactic acid fermenters can be classified into two. The first one is the homolactic fermenters and the second one is the heterolactic fermenters. So these homolactic fermenters, they use the glycolytic pathway and directly reduce almost all the pyruvate to lactate with the enzyme lactate dehydrogenase. While heterolactic fermenters, they form a substantial amount of product other than lactate. Another fermentation type is the formic acid fermentation. In this type of fermentation, bacteria it metabolizes pyruvate to formic acid and other products. Further, formic acid may be converted to hydrogen and carbon dioxide by formic hydrogenylase. There are two types of formic acid fermentation, mixed acid fermentation and the butanediol fermentation. Mixed acid fermentation, it results in the exertion of ethanol and a complex mixture of acids such as acetic, lactic, succinic and formic acid. While in butanediol fermentation, pyruvate is converted to acetone which is then reduced to 2,3-butanediol with NADH. A large amount of ethanol is also produced together with smaller amounts of acid that are found in the mixed acid fermentation. Formic acid fermenters sometimes it generates ATP while reoxidizing NADH. They use acetyl-CoA to synthesize acetyl phosphate which then further donates its phosphate to ADP. So that was about the aerobic respiration, anaerobic respiration and the bacterial fermentation, and at the end I would like to add that microbes employed different metabolic strategies and they can be differentiated from each other based on their metabolic characteristics. So all forms of metabolism, it involves chemical exchange of electron through oxidation and reduction. The transport of electrons between the donor and acceptor molecules through membrane-embedded complexes, it generates an ion gradients across the membrane. This transport of electrons is fundamental to respiration, the oxidative completion of catabolism to carbon dioxide.